everyone, and welcome to our last lecture. This week we're going to talk about the integration and regulation of metabolism, and we're also going to talk a little bit about sports nutrition. So we've talked a little bit during our second protein section, and when we talked about the feed-fast cycle, we did talk about some of the relationships between our macronutrients, and we're going to delve into that deeper today. So first we're going to discuss our learning objectives are to discuss the interconversion of macronutrients. We also want to understand how major organs and tissues interact through integration of their metabolic pathways. And we want to describe hormonal regulation of metabolic pathways to maintain homeostasis. So we'll look at this figure again at the end of this section, but I just want to touch on it briefly. And this figure is demonstrating the interconversion of the macronutrients. So basically, how are our products of our macronutrients able to be converted into one another? To some extent, we know that within limitations, interconversions of macronutrients do occur. So we know that, for instance, some amino acids can serve as precursors for carbohydrate, and we know that some TCA cycle intermediates can serve as substrates for as the carbon skeletons for non-essential amino acids. Start with the conversion of carbs to lipids. So our glucose to our triacylglycerol or triglyceride, either way. Okay, so we know that carb can be eventually converted to lipid, except lipogenesis from glucose is tightly regulated, and it requires energy, and it also requires NADPH. And you'll remember NADPH is produced from glucose with the hexomonophosphate shunt or pentose phosphate pathway. Now, one easy thing that you can remember when we're talking about the interconversion of carbs and lipids is that it's a pretty easy interconversion from glucose to glycerol, okay? So remember, triglyceride is made of the glycerol and the fatty acid, but it's the glycerol portion of it that glucose is able to be interconverted with pretty easily. So we know that, if we can produce glycerol from glucose, where are we going to get our fatty acids? Well, the fatty acids from triglycerides, when we're synthesizing them, can either come from our diet. So we've seen you are what you eat in terms of fats. Our fatty acids get incorporated into our body as they were in the diet. We can also get the fatty acids from lipolysis of our adipose tissue in order to synthesize new triglycerides. Or we can get it from our liver in the form of our lipoproteins, VLDL and LDL. Now, when we're talking about triglyceride synthesis, we know that in our liver, we have the enzyme glycerol kinase, and that is the enzyme that we need to produce triglycerides. But in our muscle and adipose tissues, we don't have this enzyme glycerol kinase. Therefore, we need to obtain our precursor for triglyceride synthesis from glycolysis. Okay, so here's a figure demonstrating that. So in our liver, we are able to phosphorylate glycerol in order to get our product G3P, and that can be used to ultimately produce or synthesize triglycerides. However, we don't have glycerol in our adipose or muscle, so we need to have our precursor DHAP from glycolysis. So here's our figure from glycolysis, where we're converting glucose ultimately to 2-pyruvate, and one of our intermediates is this DHAP, and this DHAP can be used as a precursor to triglyceride synthesis. So we just need to dehydrogenate it, and then we're at our common substrate G3P. We've talked about, so we know that carb can be converted to to glycerol, but we also know that the acetyl-CoA from pyruvate dehydrogenase complex can be utilized for fatty acid synthesis. So when we have our pyruvate and it gets broken up into acetyl-CoA and into oxaloacetate, this acetyl-CoA can be used for the synthesis of fatty acids. And we see this in this little figure here. So we didn't go too much into fatty acid synthesis in this pathway, but we did talk about this kind of little step in the box here. So when we have, when we're in the mitochondria and we have our oxaloacetate and our acetyl-CoA and they combine to form citrate, okay, so this is pretty common knowledge for us. Well, in order to produce fatty acids from this, then it needs to have, this process needs to happen in the cytosol. So the citrate can move out of the mitochondria into the cytosol, split back up into oxaloacetate and acetyl-CoA, and this acetyl-CoA can then go through an enzymatic reaction with ACC to form malonyl-CoA, which then when combined with NADPH and our enzyme fatty acid synthase can then produce our fatty acid. So it's really kind of inadvertently that carbs can be, that glucose can be converted into fatty acids. So it's an easy conversion to glycerol, and it's a really kind of indirect conversion to fatty acids. So we can see this kind of in this schematic here. So we have our common substrate, acetyl-CoA, and that can come either from carbohydrate or from amino acids, or it can come, or some amino acids I should say, or it can come from the breakdown of triglycerides. And this acetyl-CoA can then go through conversion via lipogenesis. We can get our fatty acids either from the diet or other places, and then we can form our triacylglycerol, and then this can be broken back down to our fatty acids, which then can go to beta oxidation to acetyl-CoA, and acetyl-CoA can be used for cholesterol synthesis, ketone synthesis, or it can enter the TCA cycle if we have the appropriate substrates to combine it with. Okay, so we talked about how carb can be interconverted to lipid. Now, can we go the other way? Can lipids ultimately be converted to carb? Okay, well, as we talked about, the glycerol portion can be. So the glycerol portion of lipids can be converted to glucose via gluconeogenesis. And we know that glycerol is a substrate for gluconeogenesis. This is nothing new. So here we have our glycerol, we have our glycerol kinase from our liver, we have the conversion to G3P and DHAP, and then ultimately this can be converted to glucose. And we can see this in a little bit more detail in this schematic here, where we have our DHAP from our glycerol, and then it goes through a number of reactions to produce glucose. So in summation, the glycerol can be phosphorylated and oxidized to produce DHAP, which can then be used for ultimately for gluconeogenesis. Okay, now here's where it gets a little bit tricky. So we know that the glycerol can be converted to glucose, but what about the fatty acid part of the lipid? Well, conversion of the fatty acid part of the lipid is not possible, and this is a little bit counterintuitive, but it's not possible primarily because pyruvate dehydrogenase reaction is irreversible, and we see that here. So here we have our pyruvate, and then we have the addition of a CoA. We have, it doesn't show it here. Oh, yep. We have the subtraction of a carbohydrate, and we have the removal of our hydrogens, okay, from the pyruvate. So it's a lot of things happening here as it's converted to acetyl-CoA. And we do need to notice here that this pyruvate has 3 carbons, but the acetyl-CoA only has 2 carbons, okay? So acetyl-CoA, which is our product of beta oxidation from when we are oxidizing lipids, on its own can't be used for gluconeogenesis, because once the acetyl-CoA on its own goes into the TCA cycle, both of these carbons are lost in the form of CO2 in the TCA cycle. So we really have like no net conversion of energy if we just had the acetyl-CoA that was entering the TCA cycle. It wouldn't work. We know that acetyl-CoA needs to combine with other products from from carbohydrate. And so we see that pyruvate also can be converted to oxaloacetate by pyruvate carboxylase. And this is the product that we need. So we have plenty of carbons here when we combine with our acetyl-CoA to then go through the TCA cycle. But acetyl-CoA cannot do it on its own. And acetyl-CoA can't go back to pyruvate because this pyruvate dehydrogenase complex is irreversible. So therefore, acetyl-CoA must be used for ketogenesis, cholesterol genesis, lipogenesis, or energy. Okay, now there is a little caveat to that. So that was when we are just getting our products of acetyl-CoA, which occurs when we have even number of carbon fatty acids. And that's almost all of the fatty acids that we get in our diet. However, every once in a while is an odd number carbon fatty acid, and these are partly gluconeogenic. And this is just kind of a reminder of our past lectures. So when we cleave off all of our acetyl-CoAs, we know we're left with a 3-carbon unit, which is propionyl-CoA, and this can be converted into succinyl-CoA, which is a TCA cycle intermediate. And therefore, this propionyl-CoA is partly gluconeogenic. Okay, so let's switch gears here and talk about the conversion of protein to carb. Now, we know from their lectures that most amino acids are partly gluconeogenic, which means that they give rise to a net production of pyruvate or TCA cycle intermediates, and that is demonstrated here. So we're very familiar with our conversion of pyruvate to alanine, and of course we know that pyruvate can then be serve as a precursor for gluconeogenesis. And then we also have a number of amino acids that can enter as TCA cycle intermediates. And then, yeah. Okay, so that was protein to carb. Now what about carb to protein? Well, the carb, we know that the carb, some carb metabolites can be converted to non-essential amino acids with transamination. And here is our wonderful example of our glucose-alanine cycle, where pyruvate is serving as an alpha keto acid, which combines with an amino acid to then form alanine. And this is beneficial because then the alanine can go to the liver, where it's then converted back to pyruvate, which can be converted to glucose. So in this way, the parts of a carbohydrate can serve as an alpha keto acid to produce non-essential amino acids. All right, so we've talked about carbs and lipids, we've talked about carbs and proteins. Now let's talk about proteins and lipids. So many amino acids are ketogenic, meaning they give rise to either acetoacetyl-CoA or acetyl-CoA, okay? And those can be seen here. So we know that leucine and lysine are only ketogenic, while we have other amino acids here that are both glucogenic and ketogenic. Now, this is really kind of tricky because glucogenic amino acids can be converted. Sorry, just a second. Okay. Now, we know that glucogenic amino acids can be converted into fatty acids indirectly, okay? Because they undergo oxidation to acetyl-CoA, and acetyl-CoA can then be converted to fatty acids. But this is really unlikely unless we were consuming a really high percentage of calories in our diet from glucogenic amino acids. Okay, however, that can't go backwards. So glucogenic amino acids could be converted to acetyl-CoA, which can be converted to fat. But as we talked about earlier, we can't go backwards from acetyl-CoA to pyruvate. So therefore, because the pyruvate dehydrogenase complex is irreversible. So therefore, fatty acids cannot be converted to glucogenic amino acids. All right, so after our discussion there, let's look at this whole figure again, okay, in a little bit more detail. Protein, our amino acids, can be converted to pyruvate here, Okay, so this would be our glucogenic amino acids, and they can also be converted to TCA cycle intermediates. Okay, so those would also be considered glucogenic. We also have our ketogenic amino acids here, which can be converted to acetyl-CoA, and then an acetyl-CoA can then form fatty acids. All right. Now we have, so that takes care of protein to carb and protein to fat. Yes. Okay, so then we have our carbohydrate, our kind of king that we talk about. These are converted to our triose phosphates, which is our G3P and our DHAP. So that G3P can then be used for to convert to glycerol, which can then be used for to convert to triglycerides. Also, though, it can be converted to pyruvate. Pyruvate can be converted to either oxaloacetate, and it can also be converted to acetyl-CoA. So in this way, so here we can see that we can convert it directly to the glycerol portion, but if we actually break it down to pyruvate, then it can go to acetyl-CoA and produce fatty acids, but we can't go backwards. We cannot go from acetyl-CoA to pyruvate. Okay. Then the pyruvate can then be used in the TCA cycle, and some of these intermediates can be used as alpha keto acids to produce non-essential amino acids. It's not really demonstrated well here, but okay. And then we have our fats, which are broken down, and again, the glycerol can be converted to glucose ultimately. The fatty acids can be converted to acetyl-CoA, and the acetyl-CoA can eventually enter the it can't enter the TCA cycle by itself. It needs to combine with the precursor, with substrates from carbohydrate metabolism in order to enter the TCA cycle and produce our energy. Okay, so let's talk about this major organ that has come up many times throughout our lectures. So we know that the liver, basically each tissue and organ of the body has a specific function, and that's regulated in its metabolic activity. So for instance, the skeletal muscle uses energy to perform mechanical work. The brain uses energy to pump ions against their concentration gradient in order to transfer electrical impulses. Our adipose tissue serves as a reservoir for stored fat to provide fuel to the rest of the body. And our liver, our major organ here, is the central processor and distributor in metabolism, which means it furnishes the nutrients to all the other organs and tissues through the bloodstream. And we've talked about basically how we have our hepatic portal vein that connects our intestines to our liver for the processing of our nutrients, generally. Okay, so first let's talk about the metabolic pathways for glucose in the liver. And this specifically means glucose 6P. So in hepatocytes, we can convert glucose to glucose 6P with our kinase. And in this figure, kind of our up arrows are considered our anabolic pathways, okay? So let's look at our up arrows, which means that our glucose 6P can be converted to glycogen. Now, our down arrows are catabolic pathways, which means that glucose 6P can go through our pentose phosphate pathway, or hexomonophosphate shunt, to produce NADPH and to produce our pentoses, our ribose, and then eventually nucleotides. It can also be broken down into pyruvate, which can be broken down into acetyl-CoA. The acetyl-CoA can be synthesized into cholesterol, it can be synthesized into fatty acids, and eventually into triglycerides. The acetyl-CoA, when it has the appropriate substrates to combine with, can also go into the TCA cycle. But we lose the CO2 here, so if we just had this acetyl-CoA, we would have no net conversion to energy. It has to be combined with the oxaloacetate. Okay, so and then once we get our go through the TCA cycle, then we have production of our energy through oxidative phosphorylation. Okay, next we have our amino acids. So glucose formed from gluconeogenesis can be transported to muscle for use. And new fatty acids can be metabolized in the adipose tissue for storage or used by fuel in the muscle. Okay, so our liver is a site of synthesis for many proteins from amino acids, okay? So amino acids, so let's kind of look at these and then we'll see what bases we need to cover after that. Okay, so we've got our amino acid pool in our liver. We know that from our protein section. Okay, so we know this can be converted into liver proteins. It can go into which can go into plasma, like our albumin, for instance. We know that we can have our amino acid pool in the blood, which can deliver the necessary amino acids to the tissues that need them to build proteins. And we know that amino acids can be converted into nucleotides, hormones, or porphyrins. Okay, then we know that amino acids can also be catabolized. So in this way, the amino acids glucogenic amino acids can be converted to pyruvate. But we know this requires deamination, in which case the nitrogen group goes to urea. All right. We also know that the amino acids from the muscles can be released in the form of alanine, which can be converted into pyruvate. Now, once the amino acids, the glucogenic amino acids, have been converted to pyruvate, then that can serve as a precursor for gluconeogenesis. Okay. The pyruvate can also be converted to acetyl-CoA, which can then be used to synthesize fatty acids, or it can also get broken down into our oxaloacetate, which allows us to go through the TCA cycle and also allows us to produce glucose from these intermediates in the TCA cycle. And then, of course, we also have energy production. So basically, catabolism to acetyl-CoA and TCA intermediates can ultimately result in conversion to glucose or to fat, depending on what amino acid it is, and also oxidation for energy. Okay, now let's talk about the metabolic pathways for fatty acids in the liver. So we know that we have our fatty acids in the liver. These can be used to synthesize liver lipids. They can also be delivered to the plasma via lipoproteins and also can deliver free fatty acids into the blood. Also, our fatty acids in our liver can be catabolized via beta oxidation, and this produces acetyl-CoA. Acetyl-CoA can be used for synthesis of cholesterol and ultimately of steroid hormones and bile, or it can also be used for production of ketone bodies in the blood. Acetyl-CoA can ultimately go into the TCA cycle if we have the adequate substrates for it to combine with in order to go through the TCA cycle. So therefore, fatty acids that are assembled in the liver, fatty acids can be assembled into triglycerides or released into the plasma. Most fatty acid synthesis place in the liver. And under most circumstances, fatty acids are our major fuel for supplying energy to the liver by oxidation. And acetyl-CoA that cannot be used for energy, because we don't have enough oxaloacetate, can be converted into ketone bodies, which we can use for energy. All right. So let's touch base again on our fed and fasting state. So I know we talked about this in the protein section, but I just want to talk about it kind of with more of a little bit overall whole view of macronutrients. Okay, so because glucose is the major fuel in tissues, it's really important to maintain homeostasis regardless of the timing of our last meal. Now we know that glucose so this is in our fed state, so I just ate a meal. So we know that glucose is delivered to the red blood cells and to the central nervous system to be immediately used for energy. We also know that some glucose is going to be converted into glycogen, and most glycogen from gluconeogenic precursors in the periphery. Glucose can be converted into fatty acids and stored in the adipose tissue. So that's what's happening here. Stored in the adipose tissue ultimately. So except for red blood cells, tissues can actively catabolize glucose in the TCA cycle. But in the red blood cells, we can't do that because we don't have mitochondria in order to go through our oxidative phosphorylation. Okay, so this is in the fed state. This is what's happening in the fed state. Okay, so let's talk about endocrine function in the fed state. Now, energy metabolism is primarily directed by insulin in the fed state. This is no surprise to any of us. And here's a schematic of how insulin stimulates the uptake of glucose, fatty acids, and amino acids by the muscle and adipose tissue. And you can get a little bit more detail about how this works by looking a little bit more closely at the figure here. Okay, so, but basically we have, here's the metabolic effects of insulin and its action on specific enzymes. So the take-home message is that the results of insulin are increased glucose uptake in the muscle and in the liver, increased glycogen synthesis in the liver and muscle, decreased glycogen breakdown in the liver and muscle, and increased glycolysis and acetyl-CoA production in the liver and muscle, as well as increased fatty acid synthesis. All right, let's move on to our post-absorptive state. So at this point, hepatic, so when we haven't had a meal within a few hours, basically, then hepatic glycogenolysis is the major provider for glucose in the blood. Okay, so here we have our glycogen. We don't have glucose directly from our meal at this point, so we've got to start breaking down the glycogen into glucose to be delivered to our blood and to our other tissues. So at this point, there's a diminished synthesis of glycogen. We're starting to break the glycogen down at this point. And there's also a diminished synthesis of triacylglycerols in the liver, and because we don't have as much of an effective insulin at this point. And so at this point, yes, we're breaking down glucose from the glycogen, but we're also going to start making some glucose via gluconeogenesis. So at this point, lactate from red blood cells does become an important source of carbon for gluconeogenesis. And the glucose-alanine cycle also becomes really important, where we're breaking down muscle protein into alanine, which then can go back to the liver along with lactate and glycerol in order to make some new glucose. So we're really starting to make our new glucose. So during this stage, we have less glucose from glycogenolysis and more from lactate and alanine. Okay, so in this post-absorptive or fasting state, the body, it's not as simple as just being regulated by insulin. Instead, the body relies on a variety of hormones to regulate food supply. So at this point, glucagon, epinephrine, and norepinephrine begin to have more of an important effect, and these stimulate glycogenolysis and gluconeogenesis. And this is mediated through our second messenger system. And it also activates glycogen phosphorylase in order to increase blood glucose. So basically, the net effect of decreasing insulin levels and increasing glucagon levels promotes lipolysis, activates lipase, and activates a rise in plasma free fatty acids. Okay, so here we have our graphic that's talking about how our metabolism shifts during fasting and starvation. Now, in fasting, so we haven't had food for quite a while at this point if we're fasting. We rely more on gluconeogenesis because our glycogen is depleted, okay? So basically, we have a kind of, in our last phase, our post-absorptive phase, we kind of started switching from glycogenolysis to gluconeogenesis, But at this point, our glycogen stores are pretty much tapped. So we really need to start relying more heavily on gluconeogenesis. And we do this by muscle breakdown and fat breakdown. So muscle breakdown produces amino acids that can be converted to glucose, and lipolysis produces glycerol that can be used for gluconeogenesis. So this shift to gluconeogenesis is secreted by glucagon and our norepinephrine or epinephrine, which are our glucocorticosteroid hormones. And these hormones are initiated by a response to low blood glucose. So I just haven't eaten in a while. My glycogen can't keep up. I have low blood glucose. Okay, let's bring out the big dog. Let's start making glucose via gluconeogenesis. And then at this stage 2, ketogenic amino acids are converted to ketones in order to kind of start basically, how should I say, providing energy to tissues that can rely on ketones. So it's being glucose sparing basically at that point. And at this stage, we have really large daily losses of urinary nitrogen, and that makes sense because we're breaking down muscle protein. So here we have our protein breaking down to amino acids, and then these are being used for gluconeogenesis. We also have in our adipose tissue that our fatty acids are being broken down to glycerol, and that's being also used for gluconeogenesis. Now let's move on to starvation, which we still use. So these are kind of, this is more of a gradual process. It's not, 48 hours, let's shift this. It's kind of a gradual process. So now we're moving into starvation where we haven't had food for a really long time. And in starvation, the metabolic fuel shifts again to spare body protein and conserve it in order so that the protein that we have can be used for more essential body functions such as making antibodies and enzymes and hemoglobin. I know this is a bit of a review from a couple lectures ago, but I don't think it hurts to kind of pull it all together. And so at this phase, then we really start relying less on gluconeogenesis from our muscle catabolism, and we start relying more on our fat stores as our major energy source. So we know that a lot of tissues can utilize fats just fine for energy by utilizing ketones for energy. And basically our brain can kind of start to adapt to relying on ketones as well. However, what's nice about it is that we also get glycerol when we break down fats, and glycerol is a major source of gluconeogenesis. However, we know that as our oxaloacetate levels drop and acetyl-CoA continues to be produced, then we have this really high production of ketone bodies. And again, this is a double-edged sword because we know that they have an acidifying effect on the blood, but also it does spare the need for gluconeogenesis from amino acids, so it helps us preserve our body protein. Sorry, this isn't super clear, but it is kind of interesting. So this is fuel metabolism in starvation, amount formed or consumed in 24 hours. So these are fuel exchanges and consumption, and we can see that fuel used by the brain on day 3, we're a lot heavier on using the glucose, and by day 40, we're really relying much more on ketones. Although we are still relying on glucose, and we can get that from the glycerol. Fuel mobilization, we can see that adipose tissue lipolysis we're doing throughout our starvation phase, but our muscle protein degradation is much heavier earlier in starvation, and then we have our protein sparing later in starvation. For fuel output for the liver, we can see that we have glucose output is pretty high at the beginning of starvation, but then as we move into starvation, kind of our glycogen levels are depleted, and also we're not making glucose from amino acids as much anymore. And our ketones continue to stay high though, so we're starting to rely a lot more on fat for energy. Okay, so here are your practice questions for this section. And let's move into our last topic, which is sports nutrition. So obviously for a half of a lecture, we can't get into too much depth with sports nutrition. This could be an entire course on its own, or really an entire profession on its own. But I do just want to kind of give you an overview and touch base of how this works into what we've been talking about. Okay, so potential energy from nutrients is converted to our contraction of our muscles. So basically, the amount of exercise we do, or our capacity to do exercise, depends on our reservoir of energy, which makes sense, or our reservoir of ATP. So this makes sense. And fluctuations in our body's energy demand, am I resting, am I exercising, is accompanied by shifts in the rate of catabolism of different stored nutrients. This makes complete sense. We just talked about how fluctuations in the body's environment, or basically when we're taking in food, that's accompanied by shifts in the rate of catabolism of different stored nutrients. So it really makes sense that when we have different energy demands from our bodies, we have a similar situation where our body adapts to catabolizing different nutrients differently. Okay, so the key to optimizing performance lies in strategies to maximize our levels of stored ATP that we can utilize really quickly when we need it during exercise. And we have 3 energy systems that we use during different stages of exercise. So these are the ATP CP system, the lactic acid system, and the aerobic system. And we'll talk about each of these. Okay, so first let's talk about the ATP CP system. And CP stands for creatine phosphate. Now, muscle cells utilize the high energy bond of creatine phosphate with ATP. Now, this is always kind of happening at just kind of very low rates. But when we initiate exercise, our ATP levels drop really quickly because we're just demanding so much of our body immediately. And at that very, very first stage, our bodies have the ability to utilize this ATP creatine phosphate system and how it uses it is that it replenishes our ATP by transferring a high energy phosphate from the creatine phosphate to form ATP. So this is an example of substrate level phosphorylation. So we can see this here. So we start exercising and our ATP is all converted to ADP, and then our ADP can undergo substrate level phosphorylation using creatine phosphate, and then it can be converted back into ATP. And again, this just happens right in the beginning of strenuous exercise. So 10 to 15 seconds into 10 to 25 seconds into our strenuous exercise till about, and it could go on for longer, up to 3 minutes if we're doing more of a low intensity exercise. Now, that doesn't last for long. That system is really good in the short term, but then we need to kick in a different system, and that's going to be our lactic acid system. And we've talked about our lactic acid system. We know that it's anaerobic. We know that it also undergoes substrate level phosphorylation. And this occurs when we have the incomplete breakdown of glucose into lactate, because it can't go through the TCA cycle because we don't have enough oxygen. So this is really a great system to be able to utilize if we don't have enough oxygen to produce ATP. However, in the grand scheme of things, it's actually really inefficient because we don't produce nearly as many ATP as we would if we did have the presence of oxygen. So when we do produce lactate, it can be cleared by other tissues, mostly the liver, for aerobic ATP production or gluconeogenesis. However, lactate, is lactic acid. It's an acid. It does lower the blood pH, and accumulation of this lactate can be a source of fatigue. And this lactic acid system is primarily in use during high-intensity anaerobic exercise, lasting from about 20 seconds into our high-intensity anaerobic exercise up to a few minutes. And here, this reaction we've learned about before, the conversion of pyruvate to lactate via lactate dehydrogenase. And then we have our aerobic system, which we are very familiar with from this class. So we know that carbs, fat, and amino acids can be completely oxidized in the presence of oxygen. And this is the most efficient producer of ATP. So when we're exercising, we get almost all of our energy from glucose and fatty acids. And our VO2 max, or our maximum oxygen capacity, is a really important factor in performance capacity. And that makes sense. How much oxygen can I bring into my body? That's going to determine how much oxidation via the aerobic system we can go through. And we know that this is the most efficient producer of ATP. So the more oxygen I can hold, the more efficiently I can produce ATP, the better my performance capacity. And this is the predominant system for exercise, lasting more than a few minutes, depending on the intensity. Now, all of these energy systems are always going on, and simultaneously, but they predominate at different periods in the exercise process, depending on intensity. Now, when we're at rest, our fats are our major source of energy. And really, when we're just doing kind of a low intensity exercise, like walking or something, we're going to primarily use fats for our energy. Now, during exercise, we barely use any amino acids. The only time we would do this is if we were really, really tapped. Our glycogen was completely depleted, and then we would start to break down our muscle and utilize it for energy via the glucose-alanine cycle. Now, during exercise, our major sources of energy are, no surprise, our muscle glycogen, our plasma glucose, our plasma fatty acids, and our intramuscular triglycerides. However, the extent to which we use these substrates depends on a few different factors. So first, it depends on our exercise intensity and duration, and we're going to talk about that. But basically, at lower intensities, we can utilize fat more effectively, and then as we get to be more intense, we really need to rely more on carb. It also has to do with level of training, and let's look at this carefully here, because I'm going to refer back to it in the next slides. But basically, percent VO2 max. So this is showing our energy intensity, both our energy intensity and level of training. So as we increase in our training, basically, then we increase in our ability to utilize oxygen. But basically, even no matter if you're trained or not, your percent of VO2 max is going to indicate how intensely you're exercising. So if I'm exercising pretty low, okay, well, then I'm going to be utilizing kind of all of these sources of energy. But then as we move on, okay, so let's look at our muscle glycogen. Okay, as I'm rising in intensity, I'm relying a lot more on my muscle glycogen. Okay, we know that. As I'm increasing in intensity, I'm at lower levels, I'm kind of relying very heavily on this fatty acid or plasma-free fatty acids. And then as I'm increasing in intensity, as I'm relying more on my glycogen, I'm relying less on my plasma-free fatty acids. Kind of pretty similar with our muscle triacylglycerols. As I rise in intensity, I'm actually going to use these a little bit more, but then when I get really intense exercise, I'm going to rely on them less, because I'm relying on my muscle glycogen more, and I'm also relying on my plasma glucose more, and we'll talk about why that occurs. Okay, and I'm also, extent to which the substrates are used is also dependent on initial muscle glycogen levels and carb supplementation during exercise, and we're going to talk about all of these. Okay, so at low intensity walking, I'm at a very low level of my maximum oxygen consumption. I'm not working that hard. Maybe I'm walking or something. Most of my energy is going to come from my muscle triglycerides and my plasma-free fatty acids. Okay. And then as I'm using my plasma-free fatty acids, I'm mobilizing my adipocytes, and I'm replacing those plasma-free fatty acids and then using those. So fatty acids are the favored substrate up to about 50% of VO2 max. And then when I increase in my intensity, so I'm at about 65% of my maximum oxygen consumption, such as when I'm running, then my fat oxidation does start to increase, but I have less mobilization from my adipose tissue. So therefore, I'm kind of using up what I've got in my plasma, and I'm not really able to replace it as quickly from my adipose tissue. So this could be running for like 1 to 3 hours, okay? This could happen. So this could be a really extensive process. But then when I increase a little bit to about 75% of my VO2 max, the fat, we see, okay, so we have less mobilization from the adipose tissue. So when I'm going to increase my intensity a little bit more, well, then we've kind of tapped our ability to use oxidized fat quickly enough in order to produce the energy we need for this type of intensity. And instead, we're going to shift toward carb oxidation. So this makes sense. We know when we look at the molecules of fat and we look at the molecules of carb has more oxygen than fat. So therefore, it requires less delivery of oxygen from the cardiovascular system. So it allows us to oxidize this source of energy much more quickly than we could fat. So this is when we're at a really kind of higher intensity of exercise. So our fat can keep up when we're at low intensity, but we can't keep up at higher intensity. And then we get to really high intensity, 85% of our maximum oxygen consumption. And then we depend on carb oxidation almost exclusively, or at least we rely on it much more than we did at lower intensities. So blood glucose from liver glycogenolysis and muscle glycogen become almost our sole energy suppliers. Now, at this point, we are using the glucose so quickly because we are doing such high intensity exercise that there is the potential for hypoglycemia. But our bodies do have a feedback mechanism, and actually, even though we have like decreased glucose output from our liver because we're tapping all of our glycogen stores, we also do have the ability to, at this level, this really high intensity, extensive level of exercise, we have the ability to kind of inhibit glucose uptake and increase gluconeogenesis in order to prevent this hypoglycemia. And in trained individuals, we would not necessarily have this effect. And also we can use the glycerol from lipolysis, and we can also use lactate for gluconeogenesis to prevent this hypoglycemia. And at this stage, lactate production is increased, which makes sense. We do not have, at this high of intensity of exercise, we do not have enough oxygen, so we are more likely to produce lactate. Now, the essentiality of carbs compared to fats at higher intensities is due to the need for TCA cycle intermediates in order to oxidize fat. So this is not a new concept for you guys. You know that in order to oxidize fat, we do need TCA cycle intermediates. We also know that the rate of oxidation is much slower than the rate of oxidation of carbs. Sorry, the rate of oxidation of fat is slower than the rate of oxidation of carbs. So this high intensity exercise, we need to be oxidizing a lot faster. So we're going to rely more on our carbohydrates. Also, we have limited ability of the muscle to oxidize fats. So it's able to keep up when we're at lower intensity, but can't keep up when we're at higher intensity. Now, the level of training does impact basically our ability to perform aerobically. And as we talked about, we want to be able to perform aerobically because it's much more efficient. We get much better production of ATP. So for individuals who are trained in endurance sports, we see that these individuals do have an increase in both the number and size of the mitochondria in the muscle. This makes sense. More aerobic capacity, higher capacity for oxidative phosphorylation, or energy production in the presence of oxygen. Also, individuals who are trained also have increased cardiovascular and lung capacity, so they can actually take in more oxygen, and they have hypertrophy of their type 1 muscle. And this is the type of muscle that we kind of refer to it as red muscle, and it has a really high mitochondria content and can be used more for aerobic or endurance events. So in a trained individual, we have hypertrophy of this type of muscle, and therefore we have more higher capacity for oxygen. We also know in these individuals, they have more oxidative enzymes and the increased ability to oxidize fat at high intensity. So we just talked about that the higher the intensity, the more our body has to rely on carb. But when you train more, you're able to oxidize fat more at higher intensities. And this is carb or glycogen sparing, which means that these individuals can exercise for longer at this higher capacity. And they also have an increased capacity for muscle glycogen storage, and we can see why this would be beneficial in performance as well. Okay, so when we're talking about how we can basically manipulate our nutrition intake in order to enhance performance, again, this could be an entire class or a book or whatever, but we're just going to talk about the pre-event meal here. So we want to time our pre-event meal so that we have kind of our optimum energy source during our event. So if we eat our meal way too far ahead, well, we know that's not good. When we get into our post-absorptive stage, we're starting to break down our glycogen. And we want our glycogen to be kind of at its peak when we're starting our event, because we want to be able to use all of that for our performance if possible. So we don't want to eat the meal too far ahead, but we also don't want to eat it too close to the event, because when we eat a whole meal, I'm not talking a snack, when we eat a whole meal, we know that it stimulates insulin. And when we have insulin in our system, we really don't want that when we're exercising, because what insulin is trying to do is to build up anything, fatty acids, glycogen, whatever. And what we want to do is be able to use this energy. We don't want to store the energy. We want to use it. So if we have too much insulin in our system, then it's going to lead to reduced plasma glucose. It's going to which will lead to reduced work capacity. And we know that exercise itself promotes rapid glucose uptake. So we don't want to so this is good in some situations. If someone has diabetes, we want to do this. But if we're talking exercise, basically we want to keep a balance here. We don't want to overdo it with hyperinsulinemia, because the exercise itself is already promoting rapid glucose uptake. Also, hyperinsulinemia will inhibit liver glucose output. And if I want to perform, then I want to know that that I want my liver to be able to deliver glucose into my blood, because I'm using it so quick, because I'm putting my body under a high demand, basically. And also, hyperinsulinemia could lead to muscle glycogen degradation and fatigue. So we do want to do the final meal not too far ahead, but not too close to the event. But we do want to do it at least maybe a few hours before the event, because we don't want to have basically a stomach full of food. We want to have a relatively empty stomach so that we can have rapid water absorption as well. So for endurance events, we want this meal to be high in carb but lower in fat. And we want to include low or medium glycemic index foods, since high glycemic index foods can have this hyperinsulinemic effect, that which we just discussed is undesirable when I'm just kind of starting an event where I want to have a high performance. We also want to consume an isotonic or hypotonic carb beverage 15 to 20 minutes before the event, which will give us some extra glucose but won't cause an insulin release. And for events that are lasting a long time, more than an hour and a half, then we want to be taking in glucose or a carb beverage throughout the exercise. Now, after the event, that's when we do want a high glycemic index food or beverage, because we do want this hyperinsulinemic effect, because we want to build our glycogen stores back up. We want to build our muscle back up. Okay, so here are your practice questions. And I just wanted to end with an image of food because I know that we talk a lot about the components of food, but of course, as nutrition professionals, it's really all about the food and how we can help people enjoy healthy food and help them control their disease through enjoyment of healthy food. So this is our last lecture. We'll certainly be in contact as we finish up the semester. But this is the last lecture. Just in case I don't communicate with you verbally anymore, I'd just like to let you know that I've had an excellent experience with you in this class. I hope you've had an excellent experience as well. And I know I've learned a lot. I hope that this class has been valuable for you too. So I hope everybody has a great week. Take care. Bye.